So in previous lessons, we've already learned how DNA can be duplicated and how during duplication there can be changes in the DNA sequence, what we called mutations. And recall that the key to the duplication was that the bases are sticky here. They're connected with weak bonds. These bonds can be broken and the two strands of DNA can be separated for the purpose of duplication. In this lesson then, we'll look at how it is that the DNA molecule can store information. And the general idea is that the sequence of one strand is a code for how to build a protein. So we're not talking about the stickiness of the letters, we're now talking about the sequence of the bases along one strand of DNA. That sequence is a recipe for building a protein. Before we see how DNA can store the recipe for proteins, let's remind ourselves what proteins are doing. Proteins are doing all kinds of different jobs in the cell, and they can perform different jobs because they have different shapes. So the protein that breaks the chemical bonds of glucose has a different shape than the protein that is making the chemical bond between the green and the red molecule here. And those proteins have different shapes than the ones in the membrane here that are involved in charging up ATP molecules. And those proteins are different than the channel proteins that get food into the cell or take waste out. So proteins do different jobs, and they have different jobs because they have different shapes. To take the example of this protein here, the shape of the protein influences which other molecules it can bind to or stick to. Being a large molecule, proteins are composed of many atoms, and some of those atoms are unequally sharing electrons. We saw this in the earlier lesson. The water molecule, the oxygen, is hogging electrons, so it's partially negatively charged. Well, likewise with proteins, they are going to have surface charges, and that provides opportunities for other molecules with the opposite surface charges to stick to it. In this case, the red and the green molecule can stick to certain regions of the protein. So this is why the red and the green molecules stick to this particular protein, because this particular protein has certain surface charges that are the opposites as the charges on these molecules, and so these molecules will stick to these different binding sites, and that gives the protein the opportunity to make a chemical bond, to push them together, make a chemical bond between the two. Of course, that does require ATP energy. So now, what determines the shape of a protein? Well, the shape of a protein is determined by the amino acid sequence. So here we see a sequence of colored shapes. Each of these colored shapes would be one of the 20 different amino acids found in living cells. And so a protein is simply a chain molecule. It's a sequence of amino acids, and that sequence determines how it folds up into its shape, and the shape will determine its function in the cell. In this picture, we see what an amino acid looks like. It's got a nitrogen here, it's got a carbon here, and a carbon here, and an oxygen here. So this is the backbone of an amino acid. And they differ by having a different structure for this R group. So the backbone of the amino acid is a nitrogen-carbon-carbon backbone, and what makes the 20 different amino acids different is this R group might be a hydrogen or it might be a carbon and something more complex. In this picture then we also see how amino acids can be linked together. So that link is a chemical bond called a peptide bond. So there is a sharing of electrons between this carbon and that nitrogen. So we have an amino acid here and here and here and here. And if we do a cartoon version, then we just see these little colored blobs would be the different amino acids arranged into a chain. And that chain can fold up into a shape, and that would be the protein. And incidentally, when we eat protein in our diet, in our stomach, there are proteins that can break these peptide bonds to digest the protein into amino acids. And then amino acids then can be absorbed into the blood and sent around to our cells as a nutrient. So then we're back to the main question: how could DNA be a recipe to make a protein? Well, the key observation is that both DNA and protein are chain molecules. DNA is a sequence of these bases, right? So it's a chain. The bases are linked together in a chain. And in fact, there's two strands in DNA, but we would still identify this as a chain molecule. Likewise, proteins are chain molecules. It's a sequence of amino acids linked together into a chain. So the basic idea then is that the DNA chain, this sequence here, codes for the amino acid sequence. Again, the idea then is that this chain molecule, DNA, stores the information to build this chain molecule, the protein. And we can say it again then, the sequence of DNA letters specifies the sequence of amino acids in a protein. So the sequence along one strand of DNA is the recipe to put amino acids in a certain sequence to make a certain protein. Now, you'll notice there is another strand of DNA, and it has a different sequence here. That's the complementary strand. Well, the cell has its ways of knowing which of the two DNA strands is the recipe for the protein. But that is a technical story that we're not going to tell at this point. So again, to reiterate the point, what does DNA say? It says something like this: start with this amino acid, then add that one, then add this one, then add that one, etc. So the DNA code is a code to build a protein one amino acid at a time. Now here we have the double-stranded DNA molecule shown here. And so again, the idea is the sequence of letters along one strand specifies the sequence of amino acids in a protein. And here let's consider two analogies. We'll do the 3D printer analogy first. So DNA is like the software in the 3D printer, and the printed object would be the protein then. So DNA has the information, in a sense, to make the thing real. The protein would be the object. Another analogy is a computer analogy. So just as we access information stored on a computer, the cell can access information stored in DNA, and that information is used to build proteins. In this way, then, DNA is sort of the computer of the cell. So again, the DNA molecule codes for the protein. So the sequence of DNA letters specifies the sequence of amino acids, and this chain will fold up into different proteins, and every different protein has a different recipe stored in DNA. So we can think of the DNA then as the computer of the cell. It's the repository of information to build the important protein parts. So when the structure of DNA was discovered, the scientists quickly realized that what they're dealing with here is a kind of language. And it was an unknown language in the beginning, and so the problem was to decode the language of life on Earth. Somehow this string of bases, this string of letters, means something to the cell. It means that you put this amino acid and then you link this one, then this one, this one. It means a certain protein. So the challenge was to decode the language. So let's begin to think about this problem. We have a sequence of these letters. Now there are only 4 different kinds of DNA letters that can be in some sequence here, but there are 20 different amino acids. So the question is, how can 4 DNA letters, in a sense, code for 20 different amino acids? And we can use an analogy with names. So let's say a set of parents have 20 different children. Well, you're going to want to have 20 different names to refer to those 20 kids. So the language of life needs at least 20 different names. So somehow we've got to figure out how this sequence of letters encodes different names, at least 20 different names for the 20 different amino acids found in proteins. Now at first, if you just consider perhaps each DNA letter stands for one of the amino acids, but wait a second, we only have 4 DNA letters and there are 20, so we wouldn't have enough names for the 20 amino acids. So it can't be a one-to-one relationship. It can't be that the first DNA letter in the sequence specifies the identity of this first amino acid, the second letter specifies this amino acid, the third. That won't work because we just don't have enough DNA letters. We don't have enough names to name all 20 amino acids. So what about a 2-letter code? Maybe the first 2 letters, the sequence of 2 letters here, the AT, maybe that sequence of 2 letters specifies the first amino acid, and then the AC specifies the next amino acid, and then the GG specifies the third. Well, we can do the math, and we can figure out how many possible 2-letter sequences are there. When you have 4 DNA letters, there are 16. Still not enough names for the 20 amino acids. And of course then scientists, being able to do this kind of simple math, recognized that a code of 3 DNA letters would produce enough different unique names to specify all 20 amino acids. In fact, when you do this math, there are 64 different triplets using 4 DNA letters. And if you want to see a simplified version of that math, there are 4 possible letters that can go in the first position, 4 possible letters that can go in the second position, 4 possible letters that can go in the third. 4 times 4 times 4 is 64. So a 3-letter code might work. There are 64 different 3-letter DNA sequences that could specify the 20 amino acids. It would be like parents, although they have 20 kids, they have 64 names that they refer to their kids. So some of these amino acids might have multiple names. We're going to call that redundancy. And this is, in fact, how living things on Earth use this language. It's a language of triplets. So a triplet, a sequence of 3 DNA letters. A triplet is the name for a given amino acid. First triplet specifies the first amino acid. The next triplet specifies the next amino acid. The next triplet specifies the next amino acid, and so on. And again, the cell has its ways of determining which sequence, the blue sequence or the green sequence, is actually the recipe for the protein, because certainly the sequences are different here, and they would build different proteins. So the cell will assign one of the strands to be the recipe for the protein. And so it is then that living things on Earth use DNA, the molecule of DNA then, to store the recipes for proteins. And of course, we saw earlier that the molecule can be duplicated as well. And furthermore, when it is duplicated, sometimes there are changes to the sequence. Well, if there are changes to the sequence of letters, that's going to be then influencing the recipe. So you recall that if the sequence of letters specifies the identity of the amino acids in the chain here, if you go changing these letters, you're changing the names of amino acids. You're going to be building a different protein if you mess with the DNA sequence. And so living things are using a language. And we want to think of this as a language because here we have a sequence of one kind of chemical, right? These DNA bases, these 4 letters. But that sequence means a different thing. It means a sequence of a different kind of molecule, a sequence of amino acids that build a protein. So this is the way that languages work. We have words that refer to things in the world. The word cat refers to that furry thing running around that goes meow. Well, this 3-letter sequence refers to this amino acid. And so we have a kind of a language. We've got the DNA sequence means the amino acid sequence. And why is it that all living things on Earth are using this kind of language? Well, that's because Luca had invented this kind of language. And Luca passed on this way of storing information to build the all-important proteins that keep cells alive.